Okay, so this lab gets to be a little bit different considering we're actually shifting from just kind of getting ourselves familiar with the lab and how to do stuff now with focusing on the cell itself, just like we did in lecture, but now taking a look at it a little bit closer using your scopes, right? So we're going to do the staining training itself. We're going to look at the simple stain as well as the gram stain, labs 14 and 16, and we should be able to complete them all. However, because we're going to be doing these remotely, there's a few modifications. Those modifications have to deal with what you should have access at home and any kind of warnings or any cautionary remarks just in case you need to prepare. So to make sure you can perform this lab, you need to have access to certain things. Probably the most important one of it all is access to a decent amount of water. Not a lot, but for us that are part of the lab, we get to cheat and have our little flask full of water. As long as you have, I'd say, a quart, a pint of water, gallon of water should be way more than enough. But you should also have something to use to wash it with. And that comes into item number 2, which are kind of disposable containers. What I mean by this is anything you can use, one that either paper or plastic based that you can discard because you're not going to want to keep after we keep on mixing all the stains, and it could have some bacteria in it. Not that it's harmful, but you should be able to dispose of it, right? And so I tried to look for things that could be accessible at any home, and I did a little bit of shopping on purpose just so I can look for things. And so one of the things I actually did is I bought a bunch of little Dixie plates, the ones you can buy, made out of paper. So these are the ones you can use at parties. These will work really, really well. I also found in the little kitchen aisle, you can buy these little kind of aluminum-based kind of containers so you can bake your little bread stuff, right? And then in the same aisles or in most disposable aisles, you can buy like little plastic cups and things like that. All will work really, really well. And then if push comes to shove, Ziploc bags also do the trick, right? So anything that's disposable, that way you don't have to worry about it, okay? So I'm gonna be using a couple of them so you can see how they apply really well. So these utensils will be specifically so you can catch the water, you can catch the stains and dispose of them later. The other thing you're gonna need, which was one of your requirements of part of the class, was a lighter, specifically a wand lighter, because they allow you to reach kind of across the distance, but a regular lighter will do too. Do not use an open flame. Do not use like a stove or anything like that. And then also what you need is access to any detergent, soap-based, anything will really do so you can clean your slides. And so anything from Dawn, Tide, whatever will do the trick. Just make sure you rinse it really, really well. And then most of us nowadays, because we have cell phones, we don't really need timers, but if you have like a simple timer that times it for about a minute, your regular watch will do the trick, right? As long as it has a second hand, that's really what we need to do. You should have everything you need for those. Those are the main things you want, right? So I'll switch my camera so I can show you how we're going to use this, but I also want to make sure you have the stuff for your kit. So in your kit, you're going to need specifically your main source. For those of you who remember, let me get my gloves really quick while we're doing this, is you should have had your TSB, your broth, that little tube with the red cap, put away so we can last this week, right? So I'm going to have it in my little container over here. So this guy. Now, again, remember, you're going to want to resuspend this guy. So remember how we did it before. As long as the cap is closed, you can give it a little bit of a quick shake, or you can kind of froth it around back and forth to resuspend it, right? And you should have enough material to work with it. Now, in the event that that didn't work out for you, remember, I told you to have a backup of a plate that you might have used, one of your street plates or one of the previous plates. As long as you have some organisms on it, should work. Again, no fungus. Specifically, you want to work on your bacterial colonies. So I'm going to have these both over here so I can demo them as we go. And then in there, inside your kit, so your good old guy, should have the last few things that you have remaining in there is ones we're not going to use right now. You should have one extra plate. Again, this was done so it can be spared just in case something went wrong. And then the one that should be in the baggie, which is, although it looks very similar, is a very different plate designed for antibiotic treatment. So it looks almost identical, but this is a Mueller-Hinton plate, and hence why it was in the baggie, right? So those should be on the side. Now, what we need to is we're gonna need your one of your inocula. I'll show this up close in a moment. You're gonna need the other baggie that contains the slides and the pipettes. There should be 2 large and 2 small pipettes in there. And you're gonna need that baggie that contains all the stains, all colorful stains. There should be a grand total of 6 tubes in there. And that's it. Those are all the main ingredients that we need for right now. Now, that aside, I'm going to focus on this a little bit close up. Put my disposable containers over here. Let me switch to a different camera so I can kind of go through these really quick. Let's go with this guy. Let's get a little bit closer with this one. There we go. So as you'll notice, I actually went and purchased like a little cheap kitchen towel. It's like one of the little dollar ones. So I can actually maintain the spill kit. In the labs, we would have our benches and we would actually have spill paper. It's very similar to what you use for like pets when you're potty training them. It's like plastic loaded on one side and paper on the other side so they can prevent spills. That's what spill paper is like. But since we don't have that, you can always use just a quick little towel. You probably want to have something that's disposable just so that, you know, in the event that it gets stained, you can throw it away. So again, I went by the quick kitchen aisle in the store and was able to get one for $1 too, right? So let's go through everything really quick. So I have my inoculum ready to go, nice and sterile. And my timer over here on the side. Here's my source, right? I'm going to use my other plate just in case. I'll have it one as a backup, right? And then one of the baggies that I just mentioned should have your 4 pipettes, 2 large ones and 2 small ones. Here's the 2 big ones. I'm going to put them over here. And the 2 small ones look exactly the same, just about half the size. Put them right there. And you should have access to 4 slides that you will have to wash again with some detergent and rinse out with a lot of water. Okay, so make sure that after you use any detergent on them, if any soap or any detergent is left on them, they will automatically reject the actual sample. They'll just fly off. So you want to make sure that these guys are really well rinsed. So spend a good time of an amount of water rinsing these guys out. Mine are all ready to go, so I'm actually going to use them over here so I can show you how you want to use them. Okay. And then you may need some sort of container. Like I said before, I also bought these from the store. These are the ones that hold like little Jello or like little sauces and things like that. The ones that you can have for like take home and stuff like that too. And I'm going to use my 6 little tubes over here, my Ependors. Now, I actually happen to cheat because I have one of my racks from the lab. One of those is the water. So what you're going to be looking for, let me take these guys out, and then you want to sort them out, are the following tubes. You should have one tube that literally reads 14. Okay, that's the only tube I like. It looks like a dark blue tube in there. I'll put this guy over here so I know what the number. And then the following tubes will be, will read 16, and you'll find one that says 16.1. That is this purple tube. You'll have one that says 16.2. That should have a little brown label onto it. And the liquid inside of it is brown. You should have a 16.3, which is your ethanol, should be clear. And you should have a 16.4. Should have a red cap onto it. And this is going to look a little bit more reddish in terms of its hue. And then, like I said, the last one that you probably use when you did your swabs is the one that says DW, which is the deionized water also. And again, you can find yourself any little container that you can have these inside them so that they don't spill. They work really well. So these are your main components. This is one you want to have set up, right? And so after you have your slides nice and clean, again, mine are already clean and washed and ready to go, you want to make sure you can label them. And so for that, let's introduce your Sharpie, right? And so I'm going to label one for lab 14. And all I'm really going to do is use a little frosted slide on the slide. And this may be upside down right now, but I'm going to write 14. Right? So here's slide 14. There it goes. And then we're going to need one for lab 16, which is going to be our gram stain. So same thing on the little frosted side. There's 16. That way I can keep track of them. Right? So we only need 2 for the moment. Sorry, here, let me put them up here so they can become a little bit more visible. And the other 2, I'm going to use it right now for just a demonstration. But here, that way you guys can see them facing the right way. Okay. we want to show you how do we want to actually apply the actual smears on them. So let me actually do a little bit of a close-up. I'm going to bring them a little bit closer over here. There we go. There's too much light over here, so the reflection may get to it a little bit. I'm going to disable the background so that it's a little bit easier to read. There we go. Okay, so let me bring these up over here. There we go. We have 14 and 16. And so I'm going to use one of these examples of a slide just so I can show you what we want to do. Now, I've already created one before, just so I can show you where the actual smears are going to go. So I drew a number 14. And as you can see, I just drew on it with a Sharpie just to show you. You do not have to do this. You will actually be doing that with your actual inoculum. It kind of shows you the shape of where we want to throw things. And so with a clean slide, that way I can show it to you. Let's pretend this is our makeshift 14. What you're going to want to do to lab 14 is apply 2 smears instead of 1. You can actually take advantage of the space that is present. And so you're going to want to apply 1 smear and then a second smear. Let's try to get this closer to the camera. And so once you apply them, you're going to want to spread these out really, really well, kind of like what I'm doing with this. So you're going to make 2 fairly large circles using your inoculum. And hence what I had shown you here originally. Now, a good way to do this, because your slides are transparent, is to give them just a little bit of a mark. And so you do it on the underside of the slide, not on the facing slide. So this is our slide, and on the underside, you'll notice that I'm going behind it, just where I want the actual smears to go. So I gave myself 2 little marks, as you can see, so I know where to place the actual smears themselves. So now, let me put these off to the side. Let's try doing that. First thing you're actually going to want to use is going to be one of the little small, tiny baby micropipetters over here, as opposed to large ones. Here, let me show you the difference. Clear this. Use the little baby one, right? And I'm actually going to have a little bit of water set up over here on the side, so I can do this. So I'm going to use one of my little containers. Pour it off. Just good old water. Now, accessing to DI water, this guy that came under here, kind of hard to find, right? So believe it or not, you can actually use tap water. No joke right now, I'm not trying to promote anything. So I'll try and keep the label off of this, but you can use, bottled water, right? It's still clean. It'll do the trick as well. So I just have a little bit of water over here. And what we're going to do is take the tiniest drop ever of this and apply it to our slide twice. Now you're going to see me do this. I'm going to flip this upside down so I can actually do this. But you want to be able to generate 2 tiny little drops in those spots that we marked. So 1 little baby drop and 1 little baby drop, just about 1 inch away from this guy. That's it. 2 little baby drops. Now, the reason why we want to make sure that these drops are baby, right? I'm going to do it for the second one too in a second, is because the more water you apply, the longer it's going to take for you to air dry. And you want these samples to air dry first. So if you put a boatload of water onto your slide, it's going to take forever. Now, to air dry, most slides take somewhere between 10 to 15 minutes, depending on the environment. Sometimes it may take 20, 30 minutes, so you're going to have to be patient with this. So there's my 2 little slides with 2 little drops individually. Now, we're going to want to apply our organisms. So what I'm going to do is do 2 different examples. I'm going to use one of them using our broth. I'm going to use the other one using a plate. That way you guys can get both examples, okay? You should only have to do one of them. Now remember, you're going to want to use your inoculum, right? I'm actually going to use a different one so I can use color, so I'll make it a little bit easier to notice. So here's my little inocular ready to go, okay? And we're going to apply them to each of the 2 drops. So let's do the first one. I'm going to take my solution. I'm going to resuspend it really quick. All right, make sure that the cap is on tight. That way it doesn't spill. And so we're going to take our loop. You already know your pinky technique. Just going to dip it in there really quick. Okay, close it off again. And then you'll see that I'm going to apply it to that little drop of water that we applied. And I'm going to go in big circles. We're going to smear it around the entire slide itself, or the height of the slide. And then without doing anything new, we're going to just jump to the other one and do it again. So the second smear that we're making over here is a little bit more dilute. So the first one has a heavy inoculum. The second one has a lighter inoculum. So this is your cooties. This is a little bit cooties more diluted. That's all that is happening here. Now I'm going to demonstrate this for the second one. So you just keep on using the same one. I'm just putting mine off to the side. Use my second loop to keep it clean. I'm going to use a plate. Just same thing I did before. You're going to do a quick just dab of this guy. Nothing fancy. And then same idea. Smear it all over the place. And then you can jump from this one to the next one. My slide moved a little bit, so. And that's it. You've smeared it around. I'm going to put my two guys over here off to the side. Okay. So now you have to let these dry. So normally in the lab, we actually have bench dryers. Every bench has one of these that allows you to put these on them and they'll dry them up. Since you're going to have to wait at home for this, you're going to have to be a little patient. Okay. But what's going to happen is as they dry, you'll start seeing this eventually. Let me see if I can get a bit of a close-up. As they dry, you'll start seeing they get this little white sheen on them. They will look basically dry. You want to make sure that the whole thing is fully dried out for this. Okay. So now that we've allowed our slides to dry, you'll notice that again, they'll start getting this little white sheen onto them, indicating that they're drying. It almost mimics, for those of you who've ever seen like hard water stains, like on your shower or on your car from the sprinklers or something like that. You get this kind of little white color off of them. And that's the organism kind of drying out by itself, air dried, with no other involvement. And again, remember you're taking it from just one source, twice, once onto each slide, and then you can kind of jump to the other smear just so you can thin it out a little bit more. So now that they're actually dry, then we need to heat fix them. In the lab, again, we normally have a bench heater to allow us to do that. However, we're going to do this a little bit more old school and perform this using a live flame. Now, this is going to change a little bit because back in the day, people used, your good old Bunsen burners, the same ones we just saw in the discussion, these guys. I have mine set up over here so you can see this, but most of you don't have access to Bunsen burners, or hence the modification. So I'm just kind of showing that we can actually do this. Now, traditionally, what would actually happen, I'm going to set this guy over here, is that you would take your slide as the Bunsen burner is on, and you literally do just a quick kind of do-over on top of it. You just kind of go back and forth over the flame. Now, the idea is you don't want to let it sit on the actual Bunsen burner because, one, you'd burn it and char it and then break the slide. Superheat it, breaks it down. So the easy way to do it is do a kind of a quick pass over the flame. So you just kind of do a one do-over. Now, since we don't have access to Bunsen burners at home, let me put this guy off to the side. We can use our lighter. It'll do the same job, right? And so I'm going to show you kind of using just practice one of the slides that I have, and I'll show you when we do it with those. So I'm just going to light this bad boy up. The warning, as always, don't play with flames. And all you're going to do with your slide is do a one do-over. This is all you're doing, just a quick back and forth. You can do it a couple of times. Again, notice that I'm not letting it stay on the actual slide. I'm just doing it over a couple of times. And remember, I'm doing it under the slide, not where the sample is. Okay. Now, one of the key things is that, again, you don't want to let it sit. I'm going to demonstrate it just for the sake of you not doing this. If you actually let it sit on there, you'll notice that the actual glass will get foggy and it'll get charred and it'll turn kind of nice and toasty on the underside. Obviously, you'll damage the slide. So for that reason alone, all you want to do is a quick back and forth. That flame, just going back and forth a couple of times, is enough to heat up the glass and heat fix your organisms onto the slide. So let's do that with our guys. These guys are now dry since I can actually see them visually. I can inspect them. So slide, dry, couple of do-overs, right? Once, twice, 3 times, 4 times, 5 times, 6 times, and good to go. Could do it with the other one. quick over. Again, not letting it sit there. I'm just kind of passing it over to heat fix it onto the slide. Notice that it never stays on the slide. Just quick do over. Again, under the slide, not on top of where the sample is. And that's it. You've officially heat fixed that sample onto the slide. You're actually allowing it to grab onto it. I'll put my little lighter over here off to the side. And that's our setup for actually beginning the stains. They've been air dried for a few minutes. They've been heat fixed using a lighter. And now we need to do the stains. So we'll kind of set up over here right now so we can do something. So let me kind of show you. I'm actually going to use one of the paper plates that I purchased just so I can use this as my kind of catch trap for all any spills or anything like that. Like I said before, I already have a little towel over here just in case. Okay. And then I'm actually going to use, I have my cups over here ready to go, part for it to catch the liquid. All right, so we'll use those. Again, you can use anything you want. As long as it's disposable, that's the best thing you can do. So let me put these off to the side. You can work on them. Now, again, you're going to want to have access to your water. So I have my little Erlenmeyer flask ready to go. I want to have my timer ready. I'm going to put my lighter over here off the side, move everything else so we can actually have some space on working on this. So now that we've had both of our slides air dried for a few minutes and then heat fixed with our lighter, we can actually proceed with the stains. First one we're actually going to do is lab 14, which is the simple stain. So this is, as its name implies, a very quick process. So I'm going to show you, I'm actually using my gloves to make sure I don't get any stain on me. If you want to stay away from it a little bit, you can always use something like a clothespin to hold on to this, kind of helps. And by having smaller versions of these, you can actually prop them on them so that it can actually sit on them and they'll hold them. So I'm going to look for the tube, the Eppendorf tube, that says 14 on it, right? Looks like this dark blue liquid onto it. That's methylene blue. And there's two versions on this. We can actually use our pipetter. So we can pop this guy open. And what you could do is absorb some of the fluid, draw it up, and then apply it directly over the surface of each of the two smears that you just prepared. And then once it applies the surface, I'm going to hit the timer. There's my 1 minute timer over there. There it goes. And then you could discard this. Now, I usually have a little vessel so I can toss stuff away. That way I don't have to worry about it. But believe it or not, as long as you have a container or receptacle to work with this, you can always just go the kind of old school version and just kind of pour it on top. And then you can discard the tube. So now again, you want it to sit there for a minute so that the stain gets taken in by the smear that you just produced. We're about halfway down in there, so it's about 30 seconds going. And then what you're going to want to have ready is water to rinse this out. So like I said, I already have one of these guys ready. I have my little vial over here. Okay. I'm going to take this guy out so I can show it to you. And once my timer is off, we're going to want to use a little bit of water to rinse this out. Just a couple more seconds over here. Again, you don't have to hold it. You can just keep it propped. There's my timer. And then you can actually pour it into that and then add water. Now, when I add the water over here, you're going to notice when I'm going to do something. I'm actually going to bring this up closer to the camera. Is that I don't want to actually hit the slide directly. So rather, I actually hit where the 14 is so that the water kind of waterfalls literally on top of the slide without actually hitting the sample directly. So let me see if I can change the angle over here. So I'm hitting it almost close to my thumb so that I don't hit the sample directly. If you actually end up applying the water directly to the sample, it'll just kind of wash it off directly, rather so let it kind of scurry down like a neat little waterfall. And then that's it. Your simple stain is done. You have added for a minute some methylene blue, and you wash it off with some water. Done. Pretty simple. Now, I already have some paper towels, so I can actually let this dry. I'm going to put this off to the side over here. All right. And usually I like to have my Sharpie close to it so I can kind of write what I'm doing and I know where everything's going. So I'm going to put 16 over here in a moment. Now, lab 16 is a little bit more complex. It's just going to take a couple more extra steps for it. So what I'm going to do is I'm going to rinse out, refill my wash over here. Ready to go. And I'm going to set up my lab 16. Now, same premise. Again, you could use the pipetters if you want to, or just kind of keep your life simple and go step by step. I'm going to use 16.1. This is your crystal violet. Should look a little purple in there. And like I said before, pop the cap. And then literally just kind of pour it on top of all of it. Just no need for these guys. Done. And then the rest of this you can discard, the receptacle that you have. I'm going to let it sit there for a minute. Same idea. 1 minute and each step. Keep that very easy kind of to track. Now you'll notice that the stain is going all over the place too. So when you actually hold this, this is why your gloves are more important than anything else, right? So make sure you're able to rinse out. You have some paper towels or something like that. That way you don't get it all over the place. Now, once we're done with the 1 minute stain, remember to make sure you have some water ready so you can rinse it out. Same process and protocol. Make sure you rinse it out with some water. Make sure you don't hit the actual slide directly, rather you hit the number. That way it can just kind of scurry off or waterfall off the actual slide. Got about almost 15 seconds left over here, and we'll start the next process. This is also a quick way to kind of show you why like something like a clothespin will be kind of handy. All right, a couple of seconds left. There's our timer, right? I'm gonna grab my slide, just let it kind of fall into the actual liquid. There's my slide, nice and purple from the crystal violet. And again, gonna use a little bit of water, not directly, but allowing it to waterfall. There we go. Doing an extra little rinse just for fun, just for good measure. And there's a slide. Now, if you notice, part of the smear is actually taking some of the color now. You can see a little bit of the purple crystal violet kind of attaching itself to the cells. All right, we're done with the first step, which is the primary stain. Now, step 2 is the use of the mordant, which is iodine. And so quick warning here for anybody that might have an iodine allergy. You may either make sure that you're being extremely careful, or you can actually ask somebody to help you out with that step. So iodine, our mordant, is what allows to lock in the actual first stain if it is taken in. And so you have a 2 that says 16.2. It's the one that looks brown, like iodine usually does. Right. Same idea. Cap pop. And then pour all over it. Again, same idea. You can discard this guy. And then while that's taking place, I'm actually going to prepare a little bit more water for the rinse out. And hopefully my timer. Forgot to hit that guy. There it goes. I have my watch going on too. And we're going to get another little minute to wait. Same idea. Let it sit there. And then in a minute, we'll add the water so we can rinse it out. So now what's happening is if the cell wall of this particular organism is what we now designate as gram-positive, it will actually absorb the crystal violet. And what the mordant does is that it fixes it onto it without letting it release and escape. However, if it happens to be what we call a gram-negative wall, which has that dual thin wall that we talked about in lecture, so it will not take it up. So once you actually use the iodine and the water that we're going to rinse it out, it's just all going to fall out really quick. So that's how we start differentiating between our samples. So we got just a few more seconds left. There we go. Timer, same idea. Let it scurry into it. Same concept also, quick waterfall to remove all the excess iodine. And done with step 2. I'm going to refill my water again because this one you want to be well prepared. Step 3, we have to emphasize a lot. we're going to be using alcohol, 95% alcohol, to destain what we already added. Now, the concept here is that it needs to be quick. Alcohol, as most alcohols do anyway, remove quickly most stains. So in this case, we're going to aim for about 3 seconds. You don't want to really do anything longer than that. You'll actually end up removing all the hard work that you just did. So this is a very quick destain. So you're actually going to hear me count this out loud. You may want to do the same thing when you do this too, right? So here's my ethanol. It's the one that says 16.3. It's the only one that's transparent, right? Aside from the water, obviously. Same premise. Now you're going to see me when I apply it. As soon as I add it, I'll start counting. And then I'm going to have my water ready to stop that reaction really, really fast. So this one, you have to be well prepared before you start it. So I'm grabbing my slide. I'm going to add my ethanol. There it goes. 1 1,000, 2 1,000, 3 1,000. And then immediately I come in with the water and remove it. Waterfall again. The water itself is what will stop the ethanol reaction. So discard that guy. If you let it sit any longer, more than 4 or 5 seconds, all your slide will go clear and you'll lose all your hard work. Okay. All right. So now we're ready to enter the final step of the gram stain, which is going to be the counter stain or secondary stain. This one is safranin or safranin, whoever wants to enunciate it that way. And it's the one that says 16.4. There it goes. Should look nice and deep red. And same premise. Let's do it again. Pop this guy up. Quick pour. There you go. Gonna discard this guy. I'm gonna start my timer. There it goes. Same idea. Let it sit for there for 1 minute exactly. Meanwhile, I'm going to prepare my water. To rinse it out one more time. Water ready. Timer going. About half a minute to go, and we'll be done with our steps. Again, you'll notice that my little paper plate is catching any excess stain or any spill that might have happened. And then the cup is what's catching the actual fluid. And then we'll be able to kind of dispose of that under a sink, but just make sure you run a lot of water. Most of the stuff is fairly safe, so you shouldn't have to worry about any concerns there. All right, almost getting there. Okay, my timer for the last little step. There it goes. So we'll take our slide, go into the actual cup, and then again, waterfall. Make sure you rinse everything else out. Again, never directly, just let it waterfall. There we go. May have a little bit of excess water or anything like that. This is what the paper towel is for. And there it goes. So now we have our simple stain that only add methylene blue, 1 minute under that, and rinse that with water. And now we have our gram stain slide that got hit with primary stain, which was our crystal violet for 1 minute. We washed it off. We added mordant to fix it in there if it grabbed on for 1 minute, washed that off. We hit it for about 3 seconds with the ethanol and immediately hit it with water to stop that reaction. And then we hit it with safranin, our counter stain or secondary stain, for also 1 more minute. All the steps basically 1 minute plus the 3 second destaining process with the ethanol. This process should take about 5 minutes to complete. Okay. The methylene's blue one is about 1 minute and a half. This guy takes about 5 minutes to do so. So that being done, I'll show you what the slide now looks like. It'll have a little bit of hint of colors, but now that it's ready, once you get it to dry, you should be able to look at it under your microscope or your foldscope ready to go. It's the correct size for it. Now you should be able to see the material that you collected with your broth or with your plate, depending on which one you had. Okay, so now that you have your 2 slides ready, you should have your methylene blue one, which is your simple stain. And then you'll have your gram stain that you practice from the samples, either from your broth or from your plate if you had needed a backup. Once they're dried, you should be able to put them under your foldscope to observe them and then draw from there. So we'll be able to do that over the next following days. Just make sure that you don't blot dry them like we usually do in the labs. Anything like a paper towel, anything like that will actually remove your stain. So just let them air dry. So it may take about 10, 20 minutes to do so. And then after that, make sure you can put them on your foldscope and observe them and take pictures using the actual phone adapter. That being said, you have your first 2 stains done. You should be good to go.